what that feels like. Happens all the time. You're not ready for that? You're going to have to start the introductions.
Oh man. Okay. This is Renato Kane. I'm the navigator on this watch. Any more questions about the, are we answering anything?
How about, what's one of your favorite things about your job?
Ooh. I didn't get a chance to think about that.
And
so
where
Greg,
do you live when you're not on the ship?
Greg gets to start, and then I can think about it more. Well, your name first.
Okay, your name, your seat, where you hail from when you're not on Nautilus, and one of your favorite things about your job. And on SPL so I can hear you, preferably.
My name is Greg Diffendale. I am sitting the Herc seat. I hail from Clearwater, Florida. And one of the favorite things about the job is actually, one of the favorite things about the job, Chad will be really happy about this, is that when I started in this whole engineering support side for science, I had a ship captain who basically said we're in the customer service industry, right? And so at the end of it, you want science to be happy. So that's usually a challenge to make everything work and get everything together so that at the end science is happy.
Okay. I'm already happy. How about that? It's a good start.
Oh, well, then we're done. We can go home.
I'm happy being out here.
Pack it up.
Check that box.
Time to go home.
Let's drop all this stuff off the porch and call it good. Okay, I'm ready for mine now.
Okay, you ready? Go for it.
This is Rennie again, from Wilmington, Delaware. And my favorite part is all the interesting people that you get to meet that come out here. Everyone's got really great backstories and great trajectories of where they're going, and it's kind of inspiring.
Awesome.
Paul.
Let's bring it back down the front row. Paul.
Great. Hi, everyone. I'm Paul Glick. I'm from San Diego, California, and I'm sitting in the Argus seat. I probably have two favorite things. So I'm the ROV engineering intern, so everything is new to me, and I'm able to learn a whole bunch, which is just amazing. My other favorite thing is getting to wash down the ROVs with fresh water after each dive.
That seems like it's your least favorite.
No, it's up there.
Really?
It's up there with
Why
my most
so?
favorite. It's just it's a really straightforward job, but there's it takes a while. Everything's wet. I don't know. It's
is that like washing dishes? How you just kind of can zone out?
It's kind of simple and methodical.
Yeah.
It's.
But it's cold now.
Just don't hit Mike.
Yeah.
Awesome. I like it.
We had a miscommunication. Mike got wet.
Oh, no.
Yeah, it's normally a very low pressure job, but I didn't see Mike was sitting right there behind the ROV. And
oh, no.
Yeah.
You got Mike wet?
Almost.
Low pressure.
So it was close.
Became high pressure quite.
Nice.
Awesome. Wayne?
Yeah, let's okay, now I have 0 it.
Okay.
Now that we're at point 2.
My name's Wayne Borneo. I'm from New Orleans, Louisiana.
Might as well.
I'm lead video engineer. I think one of the things I like most about working on EV Nautilus, I've been in the maritime industry for almost 20 years now, and this is my first year on EV Nautilus. I really appreciate being the opportunity to work with people who are more intelligent than I am. On most of the other boats, I'm usually one of the smarter ones on board, and I'm not challenged so much or exposed to just in an academic sense, people that just know their stuff very, very well. And there's a lot of people on this boat that know their stuff very, very well. And it's information I wouldn't be exposed to otherwise. So that's what I appreciate and like.
Awesome. I'll go next, then I'll kick to you back row. I'm Megan Cook. I hail from Friday Harbor, Washington these days by way of Boise, Idaho originally. Shout out to all the people living in deserts. I am sitting in the Science Communication Fellow seat, and I'm sitting the Lead Science Communication Fellow watch this cruise, which is a blast.
Let's put bubble back on the gauges.
Lots of things I like about my job. One of the things that I particularly
and
like
tilt up just a little bit.
is that although
Yeah.
I'm a
Okay.
biologist by training, I really like coming out on Nautilus and getting to see all these different disciplines sort of slam into one another. I like the way I've gotten to learn a lot about geology and geochemistry and archaeology and gosh, engineering and new technology development. I mean, we get to see a little bit of all of it out here, and I really like that. I make it a personal goal, but also, you know, I think it would be really hard to not come out here and learn something new every single time.
Yeah, you'd have to try very hard.
You would have to try hard to not learn. As we're doing introductions, if you're watching on the quad channel on Nautilus, in the lower left-hand image, you have the RoboCam, which is going around the control van. So come into the back row. Jamie, you want to go next?
Sure. So I'm Jamie. I'm in the data logger seat. I'm from North Carolina, and one of my favorite things about sailing with Nautilus is so I am also the science manager on board, and that means I get to train a lot of new people in science that are coming on as interns or science managers in training. And I really enjoy working with people who are having some of their first experiences coming out to sea, and seeing their passion for science, and getting a chance to share our procedure with them and tell them about sample collections and data logging and the importance of communicating all of this to the public. And I really enjoy the opportunity that pretty much every time I come out with Nautilus, I'm working with someone new who's having a pretty novel experience, and that's a lot of fun.
Cool.
Awesome. Thanks, Jamie. All right, Chad.
Hello, everybody. My name is Chad King, a lead scientist for this cruise. This is my second time on the Nautilus. I'm a marine biologist with Monterey Bay National Marine Sanctuary, which is a federally protected area, one of 14 national marine sanctuaries. I've been with them for a little over 17 years now, and this is kind of the highlight of every year, is going out to sea. The Nautilus has been just a wonderful experience, going out on Noah White ships, going out on other vessels. This is kind of why we get into this field. It's not just to sit behind a computer and crunch data and write emails. So this is definitely the highlight of the year. I'd say the favorite thing about my job, of course, is coming out, but I would also say I feel very fortunate in my particular position. I'm more of a jack of all trades. My boss was stupid enough to put me in charge of these cruises. No, I love the variety in what I do. Could be from 1 week scuba diving the Big Sur coast to doing deep sea research to developing an app where we post all of our photos for free for the sanctuary. So it takes me to many wide and varied places, and I really value the experiences it's brought me over the duration of my time here.
Chad, is your title marine biologist, or is it research
It's
coordinator?
research specialist, which definitely
Okay.
translates to more of that generalist title.
Yeah, for sure.
Usually when I tell people that, they go, "What does that mean?" I'm like, well, that's what the federal government labeled me as. So, oh, and also I hail from Gilroy, California, Bay Area native.
Awesome.
I think we got one more in the back row.
She is,
Who else is back here?
yeah, I was going to say, yeah.
Yeah.
She's
You on
not
chart?
There he is. Hello.
Hey.
Are you? Can
Alright.
you contribute to SPL?
I think I can.
Okay.
Oh.
Nope, not yet. Stand by. Are you on chart 1, 2, or 3?
I am chart 1.
Chart 1. Stand by. Okay. Okay. Try now.
How's that?
Still pretty quiet.
Not quite yet.
Is your mic like right in your mouth?
My mouth is right
Oh, there it is.
on the
There
mic.
you go.
Okay. It's a big lollipop. Right here
Yeah,
in
we
front
got you now.
Okay. I am Dane Harden,
Hey,
and
Dane.
hello, everybody. It's great to be here. It's always great to be here. My role on this cruise is to oversee sampling of deep ocean waters for analysis of what we call POPs, persistent organic pollutants. And these are materials that are man-made, that have been discharged through various human activities around the globe and have persisted for decades and decades in the oceans and accumulating up through the food webs. So we have equipment on board Hercules that will be processing, hopefully 400 liters of water through specialized media that extract these pollutants out of the water, and then we bring the media back to the laboratory and measure what has accumulated in it. So this is part of the sanctuary's ongoing efforts to understand human influences on the resources in the sanctuary and where some of these contaminants might be coming from and what we can do about it. So, and I have to say that my favorite part of the job is similar to some of the other folks here, being out here with folks who really know their stuff and are really excited and enthusiastic about what they're doing. And it's, for me specifically, it's a bit like being a detective. It's forensics. So we use the chemical profiles that we see in our samples to try to get a handle on where they're coming from and what their sources might be, and follow them into the animals and find out something about the effects. So this is my second cruise on Nautilus, and I have to say, after decades of being in this business, that the whole Nautilus crew and exploration folks really have their act together. It's just an amazing operation. So I appreciate the chance to participate.
We appreciate having you. Thank you. Could you give us an example of some of these pops?
Yeah, well,
what is one and where might it come from?
Persistent organic pollutants contain a bunch of different chemicals, and so we characterize them kind of by their family of origin.
Okay.
So one of the ones that we're interested in is polychlorinated biphenyls, which we abbreviate PCBs.
Okay.
And
you might be more familiar with that.
yeah, so these were compounds that were developed for the electrical industry back in the 1930s, actually. They were first beginning to be synthesized. And they have been used as coolants in electrical transformers. They've been used as lubricants in the marine industry. They've actually been used as extenders and carriers for the aerial application of pesticides back, in the 50s, 60s, and 70s. So they've really been distributed all across the landscape. And, they wash off the land and find their way into the ocean, and they will actually, the flux between the atmosphere and the ocean is net toward the atmosphere, so they wind up in the atmosphere, and through wind currents and condensation involved in the higher latitudes, the polar latitudes, they precipitate in rainfall and other precipitation back into the environment. And so we tend to find them in higher concentrations in polar regions than in some of the temperate areas. And
Interesting.
Historically, they're no longer being used or discharged in most situations. But as I said, they persist, and so what we're finding is that even though globally concentrations, say in the ocean at temperate latitudes, might be going down, we do find areas where, for unknown reasons, there are periodic spikes of very high concentrations of these, and Monterey Bay, unfortunately, is one of those locations. And that suggests a local source of some sort. So this effort to collect samples from the deep ocean for analysis of PCBs is hopefully going to help us determine what the sources of those are that we've been finding in Monterey Bay.
Can you tell the age of them when you collect them?
Well,
is there like a weathering?
There is a weathering process, but it's complicated a bit because well, it's some weathering. So sunlight will dechlorinate these compounds, but there's also biogeochemistry, so microbes will degrade them. So what we see in the oceans in terms of the whole broad range of PCBs, and it's a complex group of chemicals. There are 209 different individual chemicals that constitute PCBs, and we measure all 209. So when you look at the profiles of the PCBs that you measure in the ocean, they tend to be weighted toward those compounds that have fewer chlorines on them. So, and that's a combination of both the degradation that we spoke of, but also the fact that those PCB compounds with fewer chlorines are more soluble in water. So the others that have higher numbers of chlorines tend to more readily accumulate in lipids. So they're the ones that most characteristically biomagnify up the food web, whereas the lower chlorinated compounds tend to remain more in solution dissolved in the water rather than in fat or stuck to sediment particles.
That's super interesting.
Yeah. So, well, if you, I mean, I can keep talking. I'm a POP geek.
We're at 826 meters.
Okay.
We're going to 3,300 meters.
Oh, so you're telling me I've got all night to
Yeah.
talk.
You keep talking.
Okay.
You just keep talking.
I think it's interesting to hear some of the detail because I know, you know, that just, I guess, as a not in my science wearing hat or not in my Nautilus hat, just in my kind of person who lives in a community near the ocean, you know, you hear about, and I understand why, you know, that you keep certain messages very simple. You know, there are chemicals in the ocean and they accumulate and they can go from one group of animals to another group of animals as you eat them, and the higher you are in the food chain, the more you bioaccumulate. If that's a new term for our viewers, it means as you eat more and more, you are eating and digesting and moving on all the food you're eating. So those carbons are going through, but any of those toxic compounds that you pick up along the way that you are, as you eat them and ingest them, they don't move along past you. And so the higher you are in the food chain, the more of these chemicals you get. But it's interesting to hear some more detail about it.
Right. Well, so speaking of that process of biomagnifying, and this field can lead to some rather disturbing awareness of what we're doing to the planet. There is the case of what we call on the west coast of the United States the southern resident killer whale population, which is endangered. Numbers have been going down, and one of the possible reasons for that is that they have, over time, and they're apex predators in the ocean, so that They're at the very top of the food web. So, and they have lots of blubber on them. They keep warm with a thick layer of blubber on the exterior of their bodies. And when we measure the PCB concentrations in the blubber of these killer whales, the concentrations are exceeding those that have been shown to cause either endocrinological or immunological effects on harbor porpoise or harbor seals that have undergone captive feeding studies. So when in these experiments, and they're probably not doing experiments anymore where they feed the harbor seals PCBs, but when they did, they found out that at certain concentrations in the blubber, these harbor seals started exhibiting measurable health effects, and the concentration of the PCBs in the blubber of southern resident killer whales are above those concentrations now. So we can't take these big charismatic mammals, predatory mammals, into the lab and, you know, test their milk and test all their organs to see what kind of effects they're having. But it is a matter of concern. In fact, NOAA, in placing the killer whales on the endangered species list, the southern resident pod, have noted that persistent organic pollutants is one of the factors that is impeding their recovery. So efforts are being made to reduce inputs, and especially I want to shout out to Monterey Bay National Marine Sanctuary, because they've really been, based upon the information that we've been collecting in Monterey Bay, they've really been proactive in supporting the work to find out where these are coming from in Monterey Bay and what we can do about decreasing inputs. So another group of contaminants that we're measuring are, and these are legacy contaminants, they're no longer used, are chlorinated pesticides. And there's a whole bunch of different compounds. There's the DDTs, which we've all heard about. There's chlordanes. There's dieldrin. And none of these are still being used, and they haven't been used in the environment for some of them since in the United States since the 1970s. But we still find them coming off the land from river sources into Monterey Bay at masses that amount to, in some cases, 25 kilograms per year for DDT. So, and you know, that may not sound like a whole bunch, but you can have these compounds in the ocean at very, very small concentrations and have them, again, as with PCBs, accumulating up the food web and causing problems for the animals. So we measure these around Monterey Bay in that have accumulated in resident mussels. These are bivalves that live on rocky shores. And in some cases, the concentration we're measuring, and at some times exceed concentrations that have been, I guess, allowed or recommended for fish and shellfish that are being consumed by subsistence fishers. So,
okay.
And you know, we're not aware of people subsisting on these shellfish from around Monterey Bay, but we do have our own southern sea otter that lives along this part of the coast that some of them specialize on eating mussels. So there are members of the wildlife that are consuming these things and could be harmed by the contaminants in them.
So Chad, I understand, I mean, there's not a way to clean these up once they're in the water, but could you talk a little bit about, you know, what's the sanctuary's role in sort of tracking down where these are coming from and working with different stakeholders about, you know, mitigating them getting into the waters, or, you know, sort of what does the sanctuary do to?
Yeah, one of the charges of Monterey Bay, National Marine Sanctuary is to understand its own resources. And one of the big data gaps we have is water quality in the deep sea, and certainly offshore as far as we are at Davidson Seamount, which is about 50 miles offshore, perpendicular to Big Sur. This is an emerging issue that a lot of us are concerned about, not only the persistent organic pollutants, but also microplastics, which I'm sure a lot of you may have heard of. So we have a couple of projects going on, not only this one with Dane, and Dane's doing a lot of sampling in Monterey Bay too, as a part of other projects. But also we've been sampling for microplastics in the bay and as far out as Davidson since 2017. And we are working with Hopkins Marine Station and the Undergrad Research Opportunity Center at California State University, Monterey Bay, to analyze these samples for microplastics to understand how much is out there. We also have an agreement with a company that's going to be analyzing some of these plastics to understand their source, like if it's a PET plastic that comes from a water bottle. So we can kind of understand, at least with a subsample, of where these plastics and pollutants are coming from. Like you said, other than personal responsibility with things like plastics and trash, there's not much the public can do, but that is a huge contributing factor to microplastics in the ocean. The legacy contaminants, like Dane was talking about, it's really more about understanding their persistent effects, whether it comes off from rainfall through streams or if an underwater submarine canyon slide, stirs up a lot of DDT that deposited over the decades. We need to understand how that's going to be in the food chain. But this is it's hard work to do because it's remote, it's deep, and no one's ever really done too much of this kind of work before. Today we're going to be sampling, like Dane was saying. He has a pump attached to the ROV and a tube that's suspended above the ROV to sample water at over 10,000 feet deep. So not many people, and correct me if I'm wrong, Dane, has anyone sampled this deep?
No, this will be the deepest using the kind of sampling media
Yeah.
that we're using. Yep.
So we feel like this is groundbreaking work, and it's an important gap to fill in not only the sanctuary's information and understanding of its resources, but it'll be a big data point on the West Coast, I would say. And hopefully more work goes into this into the future. But like I said, and we all know, it's expensive, it's hard to get out here, and even the equipment that Dane brought, the media is expensive, it's expensive to analyze it, so it's not trivial stuff, but it's very, very important.
I might just backfill there, Chad. I tend to work not only with the science folks at the sanctuary, but also the folks that are more involved in the regulatory process. So
Right.
They are overseeing human activities around on the coastline that may have an impact on resources in the sanctuary, and they've been very supportive in helping us get the information about human activities that would be important in helping us pin down some of the sources. So we suspect certain human activities that may be affecting some of these concentrations in the near shore. And so the sanctuary works with stakeholders, helping them to understand their impacts on the environment and what they can do to minimize release of these persistent organic pollutants.
Fantastic. Several people interested in our descent rate. Greg, are we still pulling 29 or so? Awesome.
Yep.
All right, Argus is at 1156. So we'll let you do the math there at home. One free high five to whoever sends in the right answer here through the question feed. Headed down to 3,400 meters. We are already 1,160 meters, descending at 29 meters per minute. How many minutes if a train is 5 minutes late leaving the station? How many minutes to the seafloor? Send it in. hardest math problem we know a lot
5
of ROVers can do.
apples.
What time did we launch?
Vehicles were in water just a couple minutes shy of 11.
Okay.
11
Cool.
local.
So we're making good time.
23 local.
That was good. That was like the ship just came in, like power slid right into position. Everyone was ready on the back deck.
Yeah.
Mark had his coat on like an hour early. He came into the data lab, was looking around, ready.
We were ready to go, yeah.
Awesome. Go team.
I just want to note that Jamie doubted me and our ability to have everything in on time.
I'd just like to note that is incorrect, Rennie.
I would like to note on that what she just said was incorrect.
You know, she's the one doing the event log, so I think
So she
her
can log,
notes.
yeah. How do you watch the watch keepers?
How do you watch the watchers?
The watch keepers, the data loggers?
Is this like event log or it didn't happen?
Yeah, who logs the data loggers?
I was going to say, don't make me separate you, but you were literally at the far corners of the control room.
Yeah, you missed the context of last cruise when we were steadily getting more and more angry at each other.
That's also incorrect. There was no anger involved.
Okay, well, speak for yourself.
There was some fighting over the pointing stick.
That did
Oh
happen,
no.
yeah. Jamie, you were on deck out there?
Yes, I was on deck.
How was it? How's it down there?
It was really good. It was a really smooth launch.
Down there.
Yeah, that's great.
Yeah.
I was equally impressed. It's like you've been doing it all season or something.
195 days or something.
Yeah.
Something like that.
And only one more launch.
Yeah.
That was our penultimate launch.
Yeah, all goes well.
So, Megan, to give proper context, I think the reason that they said sneeze guard or shield is over here had to do with the battle with the stick of pointing and the and so there was a protective shield involved. It got vicious.
It's jousting.
Jousting. There was no jousting in the control van.
Can't get those to sync up. I think
Sorry, I had to mute. I didn't
Well, they're
actually
running
hear
at
if I was
different
supposed
baud
to
rates, I
mute.
think.
that could be. I'll do it. $23, $23400.
Oh no, they're checking the winch.
Wow. Yeah. Oh, it was just the scan speed then. See if they fix the hand. Come on.
Oh, different, different distances.
Mm-hmm. Could be. They still down there? Where'd they go?
She's there. She's you
Around
can
the
see her
side.
in the winch hold. You can just see her elbow
You can go, yeah, one elbow.
on the left.
Oh
hinge.
yeah, there she is. Future Jess.
She's wearing a wall-colored sweater.
That's it.
Yeah.
There you go, Rennie.
Winch camouflage.
Thanks, Greg. See if it stays or if one of them starts to beat it.
No, I know what I'm doing.
I mean, when I come back at 8 a.m. and they're still synced up, then I'll know.
Dan's gonna mess with everything. Don't worry.
That's true. We've got no chance.
Plus, as soon as I increase the range back on Argus, it'll disappear.
Yeah. Yep, there it goes. Slowed down just a half. 1/2 unit, 1/2 speed unit.
All right, we have another person in here on SPL. We're ready for an introduction here. We have Scott joining us, a little watching in on our watch. So Scott, want to give yourself an intro, who you are,
Oh, maybe
where
that's
you
who
hail
watches
from.
the watchers.
Scott.
That's
The one
it.
who watches
That's
in
it.
on the watches.
Yeah, you're set. And one thing you like about your job.
I get to
Yeah, give
do
yourself an
this.
intro. Come
Oh
on.
yeah, this is Scott Simon. I'm from the Marine Science Institute at UCSB. I'm the director of the REEF, or Research Experience and Education Facility. It's a teaching aquarium there. And yeah, the thing I love about my job is I get to do this.
We've had a few people come through. I'm guessing some of the students that you oversee through REEF, if I'm not mistaken. Is that right?
That is correct. A really exciting part of this is that Dr. Robert Ballard is a gaucho himself. That's our mascot there at UCSB.
Go
And we
Gauchos.
share a wonderful relationship with him. And yeah, I have undergraduates that work for me at the aquarium, and they get to come out. One student gets to come out every year as a science intern.
Yeah, we had Allie Alpin a couple of years back, or maybe that was last year.
Yeah, shout out to Allie. She's actually in Australia right now
Oh, awesome.
doing a study abroad.
She could be awake, watching
Yeah.
right now.
Yeah.
Really cool.
Alicia Dazani recently came out. She was out in the South Pacific on the Honolulu to Samoa
Yeah,
leg.
Yeah, I was
she
on
was
that.
great.
Yeah.
So got an hour and 35-minute estimation from our viewers tuned in. Paul, are they right? Is that the ballpark we're looking at?
We are now at about 70 minutes till bottom, but that's if we drove straight to the bottom.
All right.
And we won't. We're going to stop at 50 meters.
Get yourself all sorted.
Correct.
Get your game face on.
What was the estimated calculation?
Yours writing in an hour 35, which is right about right.
Yeah, it's within 15 minutes margin of error. For sure.
It's pretty good. Good math, everybody.
Squid.
Squid.
What?
Oh, it's gone.
Tiny one.
Disappearing at 29 meters a minute.
Oh. Gotta be quicker than that.
Drive by squid.
Okay.
Yeah.
We've got Science Communication Fellow Dave writing in. Hey Dave, go Gauchos. And he says what's up to Paul, who was his roommate last week. So super fun to have everybody watching on Nautilus Live right now. Do send in your questions. This is a great time to chat with our team, particularly our science team, if you have questions about what we're up to on this dive, and our ROV pilots while we're descending.
Hey, David.
So Chad, are you available to tell us a little bit more about the dive plan today?
Yes, I am. So yeah, I guess we're about 90 minutes from bottom. That's great. So this is going to be, I think, an incredible dive. I think the viewers are going to be very happy. I think we're going to be happier. It was last year, this time of year, October, in 2018, where we were coming to Davidson Seamount to explore areas of the seamount that had never been seen before, primarily for these magnificent corals and sponges that we've found up and down the slopes of the seamount itself. Now, Davidson Seamount is an extinct underwater volcano. Last erupted about 10 million years ago, so we have no fear of being consumed by lava today, which is good.
Super fine with that.
Very good, even though we'd probably be okay on the surface. And it was first explored in the late part of the century, the late 90s, and they discovered all of these amazing corals there. We visited it several times. It was such an important find and resource that the sanctuary was able to add it to our boundaries in 2008. And so now it's prohibited to basically have any bottom contact gear. There are valuable corals, precious corals down here like black corals, which in theory, when the technology reaches a point, it could be financially profitable to harvest these things. So it's a good idea to put them aside before we have any competing financial interests that would be upset about it. So the purpose was to come down to the deep part of the seamount where we had never been before to see how far these corals and sponges extended. Well, we were following a path on a very long dive last year. It was a 30-plus hour dive, much like the one today is going to be. And we had about an hour left before we were about to pull. And we had been seeing corals and sponges, not to the same densities as we see on the summit, but a lot of interesting things on the way. And we suddenly ran into a group of octopuses. Now, we had seen solitary octopus here and there. Nothing remarkable about them. It's normal to come across them.
I mean, they're remarkable, but...
They are remarkable by themselves. But we found a group of about 20 or so individuals, and as we zoomed in and landed, we realized these are all brooding mothers. And this particular genus of octopus broods their eggs. That means they take care of their eggs. They attach their eggs to rock with a cement they produce from their a special gland, a specialized gland. And they are upside down with their mouths exposed, essentially, and their arms wrapped around their mantles or their, what you would call a head, basically. The head of the octopus is against the eggs. Like, why? We've never seen anything like this. Why are they here? And as we settled down, we saw the water column shimmer, much like you would see on a hot summer day shimmering off of hot pavement. So that told us there's a really good chance that there's warm water emanating from the seamount, or from the seafloor. So immediately we kind of made two initial discoveries simultaneously. It was groupings of these octopuses that had only been seen in one or two places in the world before, and then this warm water which has not been found in the sanctuary. So we started descending down the slope. We said, wow, that group of 20 octopus were amazing. And then we subsequently ran into over 1,000 over the next hour. That's a pretty conservative estimate. It just blew our socks off. It was an amazing sight to see, and those are going to be the sights that you see throughout much of the duration of this dive. And it's just incredible to see groupings of 100, 200 octopuses within an environment of seeping water. Now, this water gets up to about 10.5 degrees Celsius, which is about a little more than 8 degrees warmer than bottom water. So it's warm to them, maybe not warm to us. But there's so many questions that we have. We're going to be measuring, like I said, at the top of the dive here, at the top of the hour. We're going to be sampling a bunch of water to understand the chemistry. We're going to be measuring temperature and oxygen to understand how those variables kind of change over time. But we don't know why the octopuses are here. Why do they like the warm water? Does it have anything to do with the warm water? Does it have something to do with the fact that the rock might be cleaned off by the venting seawater and it's easier to cement their eggs? There's a lot of other animals that are associated with the seeps too, that just adjacent to, in a non-seeping area, you see none of these animals. And these are anemones, shrimps, whelks, which are a type of snail, and other fauna. So it's just a cornucopia of animals, and we're going to have a lot of fun, not just making measurements, but also landing, looking, zooming, and just kind of figuring out what the heck is going on and why these assemblages of animals are here. So that's going to be the duration of this 30-plus hour dive. A lot of measuring, a lot of video, a lot of sampling. And for most of the end of the dive, we're going to be running transects because we haven't fully explored. This is a little bump on the seafloor off of Davidson, which is about 1 kilometer long by 600 meters wide. We've explored maybe just a small fraction of it. So we're going to be running some transects too to understand maybe perhaps how many octopus are living on this little bump on the seafloor. Is it more than what we've seen so far? Significantly more? Is it 5 times as much? We don't know. So you're going to be along for the ride today as we start counting octopuses.
That's such a good explanation. Makes me totally pumped. I just love this idea, you know, that this little knoll is 10 football fields by 6 football fields, and we just have a itty bitty little glimpse at a corner, and already such spectacular things from it. So that's a pretty big feature to cover for a little ROV. I mean, think about an ROV flying with our lights on is kind of like, you know, walking through a cornfield with a flashlight. Like you have a limited field of view while you're down there on the seafloor. So with a 30 plus hour dive, you know, we have some time to commit to checking out this landscape.
Yeah, and you're absolutely right. We're not. If people are thinking we're going to be able to literally mow the lawn and see every square meter of this feature, that's not happening. That would take many more dives. But we want to get a regular, kind of with regimented transects, kind of regularly spaced across this feature. We're hoping to have a reasonable idea of how many octopus might be here, and subsequently how many seeps are here. This is something that didn't even, wasn't even thought to exist here. Davidson Seamount's been known for a long time. We've protected it. We know about the corals and sponges, and we just serendipitously ran into this thing, which no one expected. So this also tells us that there's a potential for more of these features to exist around this very, very large mountain on the seafloor.
We just passed through 1650 meters, so that's about our halfway point. So.
And Chad, during your nice explanation, there's a watch changeover. So if you're watching at home, refresh that browser. You will see new names and faces underneath the question feed of who's on watch now.
Yeah, we got to escort the ROV down through some blue water, and now we're doing a shift change. So you'll hear who's coming on in my replacement. She's a lot smarter than me, so you'll be wowed.
We have everyone in place back here at Science for the changeover.
Was that science team was ready on the back?
Yes, we are.
Awesome. Hi, new science team. Hello.
Hi.
All right, welcome everybody. This is Scott Simon from the Marine Science Institute at UCSB, Science Communication Fellow. And we are still continuing our descent down to the Davidson Seamount. Should be there in less than an hour and a half now. And we have a new team on board. Why don't we take a moment and go around and introduce ourselves.
What are the introduction questions?
Let's see. Let me start where everybody's from.
Where do you want to start?
Let's start, well, why don't we start it with me. I'm from Santa Barbara, California, and we'll just head around the van clockwise.
Making them work. It's clockwise. Everybody's like, what's a clock? I think you're next, Wayne.
Yeah.
I don't think that's what that means.
That's easy. I'm a carryover from last shift. I'm still Wayne Borneo. I'm still from New Orleans. And yeah, it's I'm gonna take the vehicles down to the bottom and do the boring work and leave the more exciting work for the people who aren't on board as often as I am. So the girls will have more exciting time filming the octopus and the features on the bottom.
Right on, thank you. And yeah, working on a little situational awareness here. So Wayne is to my right.
Oh, right.
So as we head clockwise around the room.
We can actually show them.
There we go.
So, okay, go ahead and if you stick your hand out.
There's my hand.
And this is me.
There's
I'm
Wayne.
the video engineer. Hi. And I'll follow around as we go. You'll see the backs of people.
There we go.
There you are.
So I'm continuing on with clockwise, that's it?
Yeah.
Hi, I'm Jess Sandoval. I'm sitting in Argus right now. I'm from San Diego, California.
Right. There you go.
Michael Hannaford, Herc pilot from St. John's, Newfoundland.
I'm Erin Heffron. I'm a navigator, originally from Minersville, Pennsylvania. Now live in Portsmouth, New Hampshire.
Turn around and say hi.
Excellent.
Hi.
There she is.
There you go. Okay. And that brings us back to the back deck.
Deep, dark corners of the van.
No one's sitting at the chart table right now.
No one's at the chart table.
No.
Okay. All right. Well, let's go around to
Back to our science team, yeah?
We can start with Data in the corner.
Okay, Dana, you're up.
Dana.
Data.
Data. This is Magda Flores. I'm the data logger, and I'm from Olympia, Washington.
Okay, say hi. Wave.
Hi.
And my name's Annie Hartwell. I'm originally from Southborough, Massachusetts.
Nice to meet you.
And I'm Jennifer Brown, and I am from the San Francisco Bay Area. I live in Lafayette, California.
All right. So you're exploring your backyard.
That's right.
Yeah. Nice. Okay.
Thank you, everybody. Let's keep those questions coming.
Do we have a lot of viewers right now?
We have quite a few, actually, for this time of night, I would say.
There are folks riding in from Hong Kong, which is pretty cool, and all along the California coast. People being bummed that it's bedtime, but excited about 30 hours of diving. So they can catch it. Don't worry. Don't try and stay up for 30 hours. You can rewatch it on Nautilus Live.
Some Somebody from Wisconsin. Do a little around the world with Nautilus Live. Shout outs to Canada, the UK. We've got New Zealand on board tonight. Germany. Some people from the land down under in Australia. Turkey, Norway, the Netherlands, home of my grandmother. We've got Hong Kong, as Megan mentioned, Switzerland, Barbados, and Land of Arnold Schwarzenegger, Austria.
Oh, welcome everyone.
We are here to pump you up tonight.
Every time we go through that, I just like plan vacations in my mind. I'm like, all right, then I would like to go too.
Got it, need it. Got it.
Yeah,
been there.
Need it.
Want to go.
Go back. Jen, so what are you most looking forward to from this dive?
Oh, well, I am really excited to head back to see these different aggregations that we've already located and leave behind some of these sensors so we can learn more about them. But then to also keep looking around to see what we haven't seen yet but is there. And I just suspect we're going to find some other really cool looking aggregations and just to see what we haven't seen yet. Yeah, I guess that's the most exciting for me. And I'm going to be keeping my eye out for if we can find any of the other life stages of these octopus while we're looking around, because so far we've seen the adults and we've seen the eggs and we've seen hatchlings, but we haven't seen anything between hatchling and adult. So where do they go after they hatch? We don't know.
That's super interesting. So about how much bottom time do you feel like happened on the other two expeditions that were out here?
So after, you mean the Nautilus, between the first time we found them with Nautilus and now?
Yeah, because we had sort of a short glimpse at it, but then, you know, the community has also really been out here, which has been fantastic.
Right, so let's see, Chad came back with the Alvin and they spent some time on the seafloor. I'm trying to remember how long that was. I don't know exactly off the top of my head, but quite a few, hours.
Okay, no worries.
And then we've been back with the Monterey Bay Aquarium Research Institute 2 other times to look around and learn more about it. So some of the features that we are going to go back to today are ones that we found with the Alvin or with the Monterey Bay Aquarium Research Institute. And we have little pet names for these areas. The Octopus Garden was something that we found with the Nautilus. The Octopus Spa was first found with the Alvin, and then the last time we were here with Ambari, just in August, is when we found this feature that we're calling the vertical vent. And that's where we're hoping to start today, is at the vertical vent.
We call it the Spa Octopus.
And we call it this